This lecture corresponds to the nervous regulation of the circulation and the rapid control of blood pressure. What we're going to be doing in this lecture is going over the role that the autonomic nervous system plays a role in our regulation of our circulation, and we'll also be looking at its role in the rapid control of your blood pressure. When you look at your cardiovascular system, the cardiovascular system, its role is to be able to bring blood to tissues to deliver oxygen and nutrients, pick up waste products, and we're going to be, what drives that delivery of oxygen and nutrients is your blood pressure. So the blood pressure regulation is extremely important, because if you cannot maintain an adequate mean arterial pressure, you will not be able to adequately perfuse vessels, give them oxygen, get their nutrients. You won't be able to pick up waste products. And when you look at our cardiovascular system and how it was arranged, it's primarily arranged in parallel. So because it's arranged in parallel, your mean arterial pressure from organ to organ to organ is pretty much the same. And what's going to be important is you want to be able to regulate your blood pressure so that if for some reason there's something going on and you've got a lot of changes in local blood flow, that those changes do not significantly affect your blood pressure. So you can have massive increases in blood flow in a certain area of your body, but your blood pressure needs to be able to be adequate. You don't want those changes in local blood flow to too much dramatically affect your mean arterial pressure. But we have two systems in place for the regulation of your blood pressure. You've got neural reflexes, which involve the autonomic nervous system and even the central nervous system in some cases, which regulate your mean arterial pressure second to second. But what sets your mean arterial pressure, which I'm trying to illustrate on this dark line right here, is your renal system. Your kidneys, also some hormones, are involved in long-term regulation of blood pressure, but it sets your mean arterial pressure. My mean arterial pressure may be 93 millimeters of mercury. Someone else's may be 80 millimeters of mercury. But that is your driving pressure for perfusion of the tissues. If something happens, say for example you stood up, you stand up from lying down, your blood pressure drops very quickly. But if you've got an adequately functioning nervous system, the nervous system will bring that blood pressure back up towards normal fairly quickly. Because if your blood pressure drops too much, you're going to pass out. So the nervous system is very good at the second to second, minute to minute change in blood pressure, which may happen if you stand up, lie down, eat, sleep. Its job is to get that blood pressure back towards your set baseline. If your blood pressure increases, that nervous system is going to kick in very quickly and says, ooh, blood pressure too high, let's bring it back down towards normal. So the autonomic nervous system is going to be very good at this regulation of blood pressure. And we're going to be showing you some examples. And this is what we're going to be doing in lab. We're actually going to be doing a thing called the baroreceptor reflex in lab to show you how quickly the nervous system works to control your blood pressure. Now, if you notice this equation here, this is an equation that you must know. This is one of the equations for mean arterial pressure that you have to commit to memory. The mean arterial pressure equation, though, is just a rearrangement of your blood flow, or the flow equation, where cardiac output is your total flow. That's your Q. The TPR, your total peripheral resistance, that is your R, the resistance. The mean arterial pressure, that's your delta P. So all we did was rearrange the flow equation. So if you know the flow equation, don't really have to memorize another one. It's just the same one. We've just kind of changed the name slightly. So if you look at the equation for mean arterial pressure, two things can influence your mean arterial pressure: changes in cardiac output and changes in total peripheral resistance. So if for some reason your cardiac output drops, in order to maintain an adequate mean arterial pressure, what will have to happen to total peripheral resistance? It's going to need to come up. So we need to do something to get that blood pressure back up. So you can alter your mean arterial pressure by affecting cardiac output or affecting total peripheral resistance. When you look at the nervous system and how it controls your circulation, it's very good at some things, not so good at other things. The local control of blood flow. Autonomic nervous system, not good at it. You have areas of your circulation that are not innervated, so they can't really play a role. And even in some that may be innervated, local factors can override the effects of the nervous system. So local changes in blood flow, it won't be about the nervous system. But the autonomic nervous system is very good if you need to redistribute blood under conditions such as, for example, you need to cool yourself. I'm working outside in Texas in the middle of summer, and you start getting hot. One great way of cooling yourself is to get increased blood flow to the skin so you can lose more heat. If you're cold, blood will be restricted more to the core to help you to maintain core body temperature. The nervous system is also very good at affecting how the contractility of the heart, the pumping effectiveness of the heart, which obviously plays a role in your circulation, because if the heart's not working very well, the blood vessels aren't going to be getting their blood. And what we're going to be showing you later is how great it is at controlling your blood pressure. What I want to do is go over and review the autonomic nervous system and how it controls your circulation. In particular, the sympathetic branch. The sympathetic branch of your autonomic nervous system innervates your circulation, the blood vessels. The parasympathetics, no. Both innervate the heart. The sympathetic plays a significant role in the control of your circulation. The sympathetic branch innervates blood vessels, and it works by binding alpha-1 adrenergic receptors, and the result is vasoconstriction of those vessels. And I need to emphasize, over and over and over again, this is one of the things I want you to get down. You need to know what the effects of the sympathetics or parasympathetics on the heart or circulation and how it works. What receptors are involved, because that's really important that you know. So the blood vessels, if you have increased sympathetic activity, what happens is the norepinephrine will bind to alpha-1 adrenergic receptors, and you'll get vasoconstriction. Do you remember what will happen if I vasoconstrict a vessel? What will happen to resistance? Resistance will increase. So that's something you need to know. So if I increase resistance, what can happen to my mean arterial pressure? So remember the equation for mean arterial pressure. Mean arterial pressure is equal to cardiac output times total peripheral resistance. So if my resistance goes up, my mean arterial pressure could go up. The sympathetics also innervate the heart. They innervate the SA node, so it will have effects on heart rate. Its effects are increasing heart rate. The sympathetic branch also innervates the musculature, all the musculature of the heart, particularly those ventricles, so it will have effects on contractility. And it works. The sympathetics work in the heart by binding the beta-1 adrenergic receptors. So it's increase in heart rate. What is the fancy term for something that increases heart rate? See if you can remember what that is. What's the fancy term for something that increases contractility? See if you can remember what that term was. The parasympathetic portion of the autonomic nervous system innervates the heart, working through the vagus, It releases acetylcholine. It binds the muscarinic receptors. Now this will have effects on pretty much only heart rate. Very slight effects on contractility because it does not innervate the ventricles that much. There's some innervation of the atria, but predominantly the parasympathetics have a more important role in heart rate. They decrease heart rate. Now this picture is just trying to again illustrate the fact of how extensive the sympathetics are at innervating the visceral organs, the blood vessels, the heart. One thing I need to kind of go over again is the fact that the sympathetics will innervate arteries, arterioles, veins, but when you get down to capillaries or even metarterioles, precapillary sphincters, not innervated. They don't have metarterioles on here, but capillaries. Capillaries, no innervation. One, they don't have smooth muscle. Precapillary sphincters, most metarterioles are not innervated by the sympathetic nervous system. But those vessels that are innervated by the sympathetic nervous system, they're going to play an important role in regulation of resistance and altering blood flow. Say if I increase resistance, say to the vessels in my gut, I will divert blood away from the gut to other areas, so it can alter where blood is going. If I look at the larger vessels, the larger arteries, the larger veins, if I increase resistance, I can change capacitance, so I can alter the volume or storage of blood in those areas of the circulation. What we're going to do is start to look at the real important role of the nervous system in the regulation of your blood pressure. But we got to look at something called our vasomotor center. The vasomotor center is located in the medulla oblongata in the lower third of the pons. So you see here I've got like these three little circles, and I've labeled them C1, A1, and A2 because we're going to be looking at these areas and how they play a role in the regulation of blood pressure. You also have an area we call the cardiac center. We have what we call a depressor center and a accelerator center, but it kind of goes along with these couple of the other areas, so I don't want to really go that much into that right now. But I want to look at these areas that are found in the medulla and the pons. Now the areas are not purely anatomical locations. It's more of functional groups of neurons that play a certain role. It's not a really strict anatomic center. They're in certain areas, but no one can say definitively, oh, it's right there. It's like, oh, it's right at this spot right here. They're just kind of groups of neurons that are located. We're going to be looking at something called the vasoconstrictor area. That's the C1 area. Okay, C1 is the vasoconstrictor area. We're going to be looking at what we call the vasodilator area. That is this A1 group of neurons. The A2 area is a sensory area that receives input from a number of different places in your body. It comes up and say, oh, I'm sending information there. Then what will happen is we're going to be somehow coordinating activity with the C1 and A1 and ultimately getting effect on blood vessels or effects on the heart, which can affect your blood pressure. So I want to look at this picture here. Now this picture is what I call the wiring diagram for the vasomotor center. The circles here represent these functional areas or these groups of neurons. The A2 area is called the sensory area. C1 is our vasoconstrictor area. I want you to think of it as having this, the axons that come from it are sympathetic fibers. The A1 area is our vasodilator area. The axons that emerge from this are parasympathetic. So I'm going to come back over here and kind of go through a scenario with you. So you have afferent or sensory information coming from the periphery somewhere, going up to the A2 area of the vasomotor center. The A2 area is receiving sensory information. How active these neurons are is determined by the firing rate from these afferents. The information that can be coming to this area could be coming from, say, proprioceptors found in your muscles and your joints, which sense changes in body movement or position. It can be coming from baroreceptors that monitor changes in blood pressure. It can be coming from chemoreceptors that monitor carbon dioxide, oxygen, or hydrogen ion concentrations in blood. A lot of different information can be coming to this area. Well, A2, the A2 area sends out axons to C1, and C1 is our vasoconstrictor area. But look at what happens. Look what I have over here. It's a minus sign. A2 actually inhibits C1. So if A2 is more active, sending out firing, the firing rate increases from A2, it will really inhibit C1. It kind of is like someone is trying to run from you. You got to buy its shirt collar. It's holding it back. A2 activates or stimulates firing from A1. So this one's like, hey, I'm encouraging you, go, go, go. So if A2 is firing at a greater rate, it will inhibit C1, it will activate A1. And you notice that A1 also inhibits C1. Now C1 is that child that's always on the go. It's always run, run, run. You got to do extra work to hold this thing back. So you've got two things that are trying to hold it back, A2 and A1, because C1's always active. It has a certain firing rate. It's tonically active. It's very important in maintaining your vascular smooth muscle tone, because I want you to think again, the axons that originate from the C1 area group of neurons, those are sympathetic fibers, and they innervate your blood vessels. So you have to have a certain level of firing from C1 to maintain a certain level of vascular smooth muscle tone. Now, what will happen if you have changes in firing rate from any of these regions? Well, we'll do the first scenario. C1. If C1 is more active, that's firing, 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 what it'll do is it's going to increase all of these three things. So let's figure out how it works. Sympathetics innervate your heart. They increase cardiac contractility, therefore stroke volume will go up. It innervates your cardiac conduction system. It increases your heart rate. So both of these two together will result in an increase in your cardiac output, since these two multiplied together is equal to your cardiac output. So if you increase your cardiac output, you will increase your mean arterial pressure, since mean arterial pressure is equal to cardiac output times total peripheral resistance. If the firing from your sympathetic decreases, there's less firing, you're just going to get the opposite. Drop in stroke volume, a drop in heart rate, therefore cardiac output drops, and then mean arterial pressure will drop. If you have the changes in firing from the parasympathetics, which also innervate the heart, you will have, if you have increased firing of parasympathetics, it decreases heart rate. You notice I did not write or show you decrease in stroke volume, since it doesn't have a huge effect on contractility. But because it decreases heart rate, it could decrease cardiac output, since that's part of your cardiac output equation, and therefore decrease your mean arterial pressure. If you decrease firing of the parasympathetics, heart rate goes up, cardiac output goes up, and therefore mean arterial pressure goes up. Remember, both sympathetics and parasympathetics innervate the heart. How fast that heart's going is determined by both how active the sympathetics are, how active the parasympathetics. think of the parasympathetics as your brake and your sympathetics as the accelerator. So remember, I had described in class is if I want to speed up the car, I can either let off the brake and or push down the accelerator. If I want to slow down my heart, I push down the brake or let off the accelerator. So this is what you're seeing here. But remember, the sympathetics also innervate your blood vessels. And so if C1 is more active, besides its effects on the heart, it can affect blood vessels. So if it's more active, we'll see what happens up here. If it's more active, sorry, didn't have my thing on there. Doesn't have it written in there, sorry. If you have increased C1 activity, you will have increased firing rate. You will vasoconstrict. I don't have that in here, but you will vasoconstrict. If C1 is less active, you have decreased firing rate, blood vessels will dilate. Okay, blood vessels will dilate. So why do they call the A1 area of the vasomotor center the vasodilator center? This is why. A1, since it inhibits C1, blood vessels are going to dilate. So remember, if I have decreased firing from C1, what will happen is blood vessels will dilate. Now, what will happen to mean arterial pressure in either of these two instances? If you constrict a blood vessel, which would happen up here, if you constrict a blood vessel, resistance will go up. Resistance goes up, mean arterial pressure goes up. This scenario here, if you have decreased firing rate, vessels will dilate, resistance will drop, mean arterial pressure will drop. So what you're seeing are, especially when you're looking at the C1 area of the vasomotor center, what you're seeing is you can change mean arterial pressure by changing both cardiac output and total peripheral resistance. We will revisit this diagram when we look at specific scenarios of what, and one of them we'll be doing in lab. So I'm going to revisit this again and give you some scenarios. The vasomotor center is also innervated by higher nervous centers, not just getting information from these chemoreceptors and these proprioceptors or even the baroreceptors from the periphery, is you also have higher brain centers which can influence that vasomotor center. Just think what happens when you're anxious. If you have anxiety, do you notice that things may happen to your blood pressure or your heart rate? If your hypothalamus has a lot of roles in temperature regulation, it plays a role in thirst, in food intake, anger. A lot of those things influence your blood pressure, influences your heart rate. You think about fight or flight response. The hypothalamus also plays a very important role in your fight or flight response. So a lot of different areas or different things can influence your blood pressure by way of the vasomotor center. If you're in pain or certain emotional states, which you're usually working through the cerebral cortex, you can get changes in blood pressure. Now the effect of norepinephrine in the adrenal medulla, we're going to be looking at that when we talk about the mass sympathetic response. So you already know norepinephrine's role because it's released by the sympathetic nerve fibers, but also we're going to talk about that norepinephrine and epinephrine are released by the adrenal medulla in a mass sympathetic response, and we're going to see what its effects are. Now there is a thing called a sympathetic vasodilator system. But a lot of this is, what I'm going to say, in humans is very skeptical about this. In cats and dogs, they have studied this vasodilator system a little bit more in depth than humans, and they've been shown to this system is discharges in response to things like fear, rage, apprehension. But in humans, the role of a sympathetic vasodilator system is kind of still unclear. They say that it's probably responsible for an initial increase in blood flow that happens, right when you begin to exercise or right before you begin to exercise, and it's mediated through the anterior area of the hypothalamus. This is my abbreviation for hypothalamus. It's also an abbreviation for hypertrophy, but this is my abbreviation for the hypothalamus. When you exercise, mainly your changes in blood flow that happen in the muscles that you're working during exercise is because of local factors that are being produced, like lactic acid, carbon dioxide. But this, they say, it's involved in initial, before you can even start building those things up. It's like, let's start to increase blood flow to here. Now these vessels, or this sympathetic vasodilator system, the fibers are cholinergic fibers that release acetylcholine. But its role in humans is still unclear, because what the research they've done is that vasodilation that occurs at the start of exercise is not very constant, it's not very marked. But this is what they think its role in humans is maybe just at the initial onset of exercise. So these are really, actually, believe it or not, sympathetic fibers that happen to have actually cholinergic instead of adrenergic. This system may play a role in something that we call the vasovagal syncope. But what I'm about to talk about are something called the defense. You have a defense and a depressor area in your hypothalamus, because your hypothalamus does a lot for you, believe it or not. It plays a very important role in a number of different things. But you have two distinct areas in the hypothalamus. They have different effects. And what I'm going to talk about is I call playing possum. Playing possum is mediated via the depressor area. And this area is going to be involved in what we call vasovagal syncope. So you may have encountered or be one of those people that a certain thing, certain trigger, will cause that person to pass out. And so the syncope is like the passing out. And vasovagal kind of tells you kind of how it's working. Somehow through vessels, vagal, like, what does the vagus do? Well, with vasovagal syncope, there's some sort of trigger for these people. What results is you have a cardioinhibitory response, and you may also have a vasodepressor response. So a cardioinhibitory response is heart rate would drop, and you'd have a decrease in cardiac contractility. A vasodepressor response, you would have a drop in blood pressure, but not necessarily have a change in heart rate. People with vasovagal syncope may have a mixture of a cardioinhibitory response and a vasodepressor response. So what you'll see with these people is their heart rate drops, contractility drops, enough that you're not going to get enough blood out of the heart. Not enough blood out of the heart going to the brain, you're going to pass out. Same if your blood pressure drops. If your blood pressure drops, you're going to pass out. So they pass out because of significant hypotension in bradycardia. And somehow the trigger working through the cortex sends information to this depressor area of the hypothalamus, working through the vagal centers in the medulla, goes off to the spinal cord into working through the blood vessels and the heart. The people are going to pass out. So I call it playing possum. So if you think about possum, they play possum. They're not dead. They look like they're dead because their heart rate's really low. Their blood pressure's real low. Not that I've personally gone to take a blood pressure off a possum, but I'm sure that's what happens. Now the vasovagal syncope is seen, or the trigger for this is, there's a huge range. You can Google it. It's amazing what triggers are for certain people. Most people think about, oh yeah, the sight of blood makes certain people pass out. That could be a trigger for some people. Just strong emotions can be a trigger for it. There are some people who have vasovagal syncope that's triggered by going to the bathroom. Either, defecation or micturition. Micturition is just a fancy term for going pee. So that's amazing when you have someone that has it just from going to the bathroom. So there's a large range of people with vasovagal syncope. The biggest problem with vasovagal syncope is the injuries that you sustain by when you pass out. The defense area, though, of the hypothalamus has a completely opposite effect. The defense area is like, oh yeah, bring it on. So defense area is like, I'm not going to pass out and hopefully someone walks away and says, okay, I don't want to mess with you. Defense area is like, okay, I'm standing up to you, I'm going to fight you. So what's going to happen? Your heart rate's going to increase, your blood pressure is going to increase. So that area mediates completely opposite effect. So the vasovagal syncope is working through the depressor area of the hypothalamus. All right, here comes the big stuff. Looking at the nervous system in the control of blood pressure. So, and again, short-term regulation of blood pressure. So the first thing we're going to look at is something called the mass sympathetic response. This will come into play when we talk about exercise, because exercise is an example of a mass sympathetic response. But it could be something like if you're hemorrhaging, or somehow you have a hard time maintaining your blood pressure. A lot of things are entailed when you talk about a mass sympathetic response. So when someone says you have a mass sympathetic response, what they're talking about is you've got the sympathetic nervous system working through changing or affecting the heart. You're also affecting blood vessels. You're going to have vasoconstrictor effects. You're going to have heart effects on the heart. What you have is sympathetics effects directly on the circulation in the heart, but also the sympathetics send out axons down to the adrenal medulla and cause you to put out a lot more norepinephrine and epinephrine. So the adrenal glands also put out these hormones, which then have effects on your circulation in the heart. So we're going to fill in this diagram. And again, it's very, very important that you distinguish between what receptors are involved. So if you've got sympathetic stimulation, norepinephrine is your neurotransmitter. We can fill in here where it should say alpha-1 adrenergic receptors. Those are going to be found on your vascular smooth muscle, in your blood vessels, okay, in your blood vessels. So what it will do is constrict the blood vessels, which would increase your blood pressure by increasing resistance. It works on beta-1 adrenergic receptors found in your heart. And the beta-1 adrenergic receptors, the effects would be to have a positive inotropic and a positive chronotropic effect on the heart. So this means you increase cardiac contractility. This one here increases heart rate. So if you increase stroke volume, you increase heart rate, that would increase your cardiac output, which could increase your mean arterial pressure. Well, the epinephrine released by the adrenals also bind to alpha-1 adrenergic receptors on your blood vessels, and at high levels will constrict your blood vessels. Epinephrine binds to beta-2 adrenergic receptors. Now these are going to be found in what I'm going to refer to as your protected circulations. Blood vessels that have beta-2 adrenergic receptors, they will dilate in response. So think about if you are exercising, which is a mass sympathetic response. Do you really want to constrict blood vessels to your heart? Nope, don't think so. I definitely want to make sure those vessels are open. So you have a lot more beta 2 adrenergic receptors on your coronary blood vessels. But a little side note, the coronary circulation is predominantly controlled by local factors. There's not a lot of innervation to begin with, and even then, you don't have a lot of adrenergic receptors. So, but you do, of the ones that you have, you got beta 2s, and so they dilate. You'll see this in, you'll see a little bit more of this in your skeletal muscles too. You will also see this, things like the liver is where you can see more of the beta 2 adrenergic receptors. So if you think about exercise and you think about stress, what type of things do you want to happen? And we're going to be spending a lecture on this too. When you exercise, you want cardiac output to go up. And so how are you going to be doing that? How's the nervous system going to be working to do that? So you know its effects on the heart. You need to also increase venous return, and that's going to be working by constricting larger blood veins. That's going to divert blood or borrow blood from the venous system and bring it back to the arterial system. If you bring it in back to the heart, that blood eventually becomes your cardiac output. Venous return becomes your cardiac output. You are going to constrict arterioles, which would affect resistance and affect blood pressure. It's kind of important when you're exercising where you can have such increases in blood flow to your muscles that if you did not have your nervous system, your blood pressure can drop significantly because that massive increase in flow in those tissues. Nervous systems can play a very important role in maintaining your blood pressure during severe exercise. When you think stress too, same type of things. When you're stressed, fight or flight type of things like that, same type of things are going to be happening. You're going to be constricting vessels, resistance is going to come up, mean arterial pressure can go up. You're also going to have changes on your heart also. Now, what I want to do is talk about a really important mechanism for maintenance of blood pressure. And this next mechanism will be involved, or what we're going to be actually doing in lab. And we're going to be looking at this baroreceptor reflex. These other ones we will discuss, but this is the one that we're actually going to be doing in lab. So this is a good time for me to take a break because I'm getting tired and I have to get these cats to be quiet. So I'm going to take a break. Next, this is part 1. Part 2 will go over the baroreceptor reflex and these other reflexes. I'm going to spend a lot more time on the baroreceptor reflex since we're going to be doing that in lab.